clusters of pacemakers, the node and the atrioventricular node. And together, those work to allow our atria to depolarize and then our ventricles to depolarize. So this morning in lecture, we had talked about the action potentials in both of these. We looked at the action potentials in the pacemakers, and then we were ended lecture by looking at the action potential in the cardiac muscles. So if you guys remember, this morning, we had an action potential that looks something like this. Potential. Small here. So we had our membrane potential over time, and it looks something like this, where we have an influx of sodium, a little bit of potassium, calcium channels open, and then we sort of have this drop back down like this. So our action potential in cardiac muscle cells looks a little funny, and it's because specifically it looks funny because of this area right here, right? That plateau that we see. Now, we said that the significance of that plateau was to essentially elongate the action potential to make it take longer. And what that does is it prevents cardiac muscle from going into tetanus, which is something that we definitely want to avoid, right? And that's kind of where we had left things this morning. But the plateau was due to the flow, essentially the outflow of potassium and then that inflow of calcium that we were seeing, right? And this is different from a neuronal action potential that you guys looked at in 50A, where you had sodium and potassium, right? That was what we were used to from neurons. But here we introduce calcium into the mix, and what that does is it sort of elongates the action potential a little bit. Okay. So really important functional significance, right? We don't want the heart to go into tetanus, so we have to make a long action potential so that we can't stimulate the muscle so fast when we go into tetanus. But what we also want to do is we want to look at the role that this calcium plays in the contraction of the muscle. Right? So that's kind of what I wanted to do to just sort of finish a few thoughts from this morning. I wanted to go ahead and look at the role of this calcium in the actual contraction of the muscle. Right? So let's go ahead and draw a cardiac muscle cell. Right? We have our cardiac muscle cell that we looked at on Wednesday. This sort of branched fiber that we see right here. We've got our nucleus, our striations. Right? Striations of our cardiac muscle cell that we have on Wednesday. So let's also think back for a minute. Let's think back to what we know about calcium and muscle contraction from skeletal muscle, right? So what role does calcium play in skeletal muscle contraction? Do you guys remember? I know this is like asking you to dig deep into the memory banks, right? But what role did calcium play in skeletal muscle contraction? Exactly, right? So remember that for skeletal muscle, we have a motor neuron releasing acetylcholine onto the muscle, right? And we start the action potential in the muscle because that acetylcholine opens sodium-potassium channels. You have that flow of sodium and potassium, you depolarize the muscle. And the calcium is stored where in the skeletal muscle cell? What's that? It's not Jesus. That's not the right name. That's not the right place. Sarcoplasmic reticulum, though, is the right place, right? And it's the sarcoplasmic reticulum that stores the calcium. Okay, so we have a sarcoplasmic reticulum, and when we start that action potential, what it does is it causes the sarcoplasmic reticulum to dump all of that calcium into the sarcoplasm, and it binds to troponin, moves tropomyosin off of the myosin binding sites, and then So let's go ahead and take a look at the cardiac muscle cell membrane. So what we're going to do is we are going to take a little section of membrane right here, and we're just going to go ahead and blow it out. Right? So we're going to take a closer look at that section of membrane. We remember maybe that skeletal muscle has not just a flat membrane, but it has these sort of indentations like this that were called T-tubules. You remember T-tubules? Cardiac muscle also has T-tubules. And cardiac muscle also has sarcoplasmic reticulum. Okay? So in our cardiac muscle as well, we're going to find sarcoplasmic reticulum. And in that sarcoplasmic reticulum, you're going to find calcium. Okay? So there's calcium in there as well. So we're just going to go ahead and make Ca red dots. Kind of a difference that we haven't gotten to yet between skeletal muscle and cardiac muscle is that the sarcoplasmic reticulum in cardiac muscle is a lot smaller than it is in skeletal muscle. It's not as big because we don't store as much calcium in cardiac muscle. Okay? So it's a little smaller, there's not as much, it's about 10% smaller. So the sarcoplasmic reticulum is about 10% smaller in cardiac muscle. We still need calcium for myosin and actin to interact with each other. And just like in skeletal muscle, cardiac muscle has troponin, tropomyosin, and actin and myosin. So it has those regulatory proteins as well as those contractile proteins. Okay. So what's going on here, right? We know that we have calcium flowing across the membrane, right? So what happens with cardiac muscle is a little bit different. Again, remember with skeletal muscle, it's just acetylcholine and sodium and potassium that start the action potential, and then we dump all of that calcium from the sarcoplasmic reticulum. With cardiac muscle, it's a little different because here we have calcium flowing into the cell extracellularly, and we see that on the action potential. So the calcium that is extracellular is coming into the cell through calcium channels. Coming into the cell through calcium channels like this. So we have that extracellular calcium that comes in and it does this. Okay? And then what that does is it triggers the sarcoplasmic reticulum to dump its calcium into the sarcoplasm, and then the muscle contraction proceeds as you guys have learned it in the past. Okay? We have the calcium binding to troponin. Tropomyosin is moved out of the way so that actin and myosin can bind with each other, and the sliding filament theory happens as you've learned previously. Okay? I'm not going to bore you by going through all of that again, but it's just at this point it's the same. The biggest difference is that we trigger the release of calcium from the sarcoplasmic reticulum by having that extracellular calcium. So cardiac muscle and skeletal muscle are a little different in that regard because skeletal muscle does not rely at all on extracellular calcium. Okay? But cardiac muscle, the heart, does rely on extracellular calcium. Okay? It has a functional role because it works to help us prolong the action potential, but it also is necessary for the contraction of the cell. So this is why when we have patients with heart issues, with cardiac issues, that we do blood work to look for calcium levels in the blood, among other things. It's not the only thing that we're looking for. But when we have heart issues, one of the things that we're looking for is calcium levels in the blood. Because that extracellular calcium is necessary for contraction, if levels get too low, we might have difficulty contracting cardiac muscle, right? That would lead to a slowed heart rate and potentially a heart attack issue, right? Whereas if calcium levels are too high in the blood, that can lead to the opposite sort of a situation where the heart becomes hyperexcited and the heart rate won't slow. Right? We call that tachycardia. And that's not a good situation either. So when we have patients with heart issues, one of the first things we're going to look for is we're going to look for blood calcium levels to make sure that those are correct and that the extracellular calcium is doing what it should do to help you achieve the contractions. Okay? All right. There, that's what I didn't get to in lecture this morning. All right. Are there any questions about that? And again, I know this morning's lecture was massive. There's a lot of material in there. So take the weekend to go through both the action potentials of the pacemaker cells, the action potentials of the cardiac muscle cells. Let me know if you have questions on Monday. Email me Monday, I guess I won't see you, but let me know if you have questions. Okay. Any questions about any of that? Okay. So now we've looked at kind of what's happening at the, I guess, molecular level of the heart. And now, tired of my allergies going. It's not helping. All the trees are still too happy. Now we've looked at kind of what's happening in the heart at sort of a molecular level, if you will. We spent a lot of time looking at action potentials in these spaces. We just looked very briefly at how the heart relies on extracellular calcium channels to contract. Now, what we want to do is we want to take what we know about the heart and the heart's function, and we want to look at the heart as a whole. Okay? Because looking at the heart's contraction as a whole is what's probably how we're going to use most of this knowledge kind of move forward. Because if we have somebody going into the emergency room with heart issues, we are going to want to look at blood work for things like blood calcium levels, but we're also going to want to look at the conduction of the heart. So let's take a look at how we can use some of this knowledge to look at the conduction system in the heart. So again, back to our diagram from this morning, right? We know that we have the sinoatrial node. We have an action potential in that sinoatrial node that will spread through gap junctions to the atrium, okay? So it goes from here, over to here, and it goes all the way through the atrium. Eventually, that sinoatrial node will affect the atrioventricular node, okay? Remember, these two have slightly different rhythms from each other, but because the sinoatrial node is essentially upstream of the AV node, it sets the rhythm for the whole heart. That's why we usually call it the pacemaker of the heart, because it does set the rhythm for the entire heart. Okay. So depolarize the AV node, and then through gap junctions and through this nerve network that we see in the heart, we're able to spread that action potential through the ventricles. So we have that action potential traveling down the bundle of His, the bundle branches, and ultimately around to the Purkinje fibers. Okay. So we know, and you guys have told me this already, you told me this on Wednesday a couple of times, we know that when a muscle cell is stimulated or depolarized, what it's going to do next is it's going to contract, right? So we know that the contraction follows. So if the depolarization is essentially, starting here and working its way in this direction, we know that the contraction is going to follow the same. Right? So let's go ahead and take a look at what we call the cardiac cycle and look at the different phases of the cardiac cycle so that we can understand what's happening when the heart contracts. So the cardiac cycle is just kind of following those contractions of the heart. But now we have blood in the ventricles, so we can see what's happening with the blood. There's a couple of terms that we need to be familiar with here. We're looking at essentially a contraction relaxation cycle. But the terms that we use for those are a little different. For relaxation, we use the term diastole. And for contraction, we use the term systole. So we have relaxation and contraction. Okay? So we're alternating between a cycle of relaxation and contraction, or a cycle of diastole and systole. Diastole and systole. You guys have seen these terms before. You know you have, because you probably either measured blood pressure or you've had your blood pressure measured. Right? When you have your blood pressure measured, you're usually given two numbers. You can do it as one, but we usually do it as two. What are those two numbers called? Your systolic and your diastolic blood pressure, right? So those two terms have everything to do with what we see right here. Right? Next week, when we look at blood vessels, we'll take a look at blood pressure and see how systolic and diastolic blood pressures correspond to systole and diastole, right? But there's definitely a connection there, and we'll look at it next week. You probably have seen it as well, like on a, I don't know, like medical TV show, or maybe there's a movie where they rush somebody into the ER. I don't know, what do we even watch now? Is Grey's Anatomy still a thing? Is it no? I don't know. Is it even about medicine? It's still on though, right? Or is it over? It's not a thing. They don't do medicine on it anymore. Now it's just drama, huh? Yeah. They have like a minute of medicine and the rest is drama. I guess they just work in a hospital. I always liked House myself. That was my show. Yeah. Greg House is like my kindred spirit, so I liked him a lot. But nevertheless, you probably have seen a TV show or a movie where they've rolled somebody in on the gurney into the ER, and they say, our patient is in asystole, right? We've probably all seen that, right? What does it mean to be in asystole? So systole is contraction of the heart. A, what does that prefix A mean? Nothing, right? A means without, right? So if you're in asystole, it means that you are without contraction, right? The patient is in asystole, it means their heart's not beating, right? Their heart is stopped, right? It's right before they put the paddles on their chest, right? To put an electrical current through their chest in an attempt to restart the action potentials in the sinoatrial nerve. Right? Okay. So we've seen these terms before, but we have to get used to using weird terms, I guess. We can use these terms for either chamber. So I can talk, for instance, about the atria being in diastole or systole, okay? Or I can talk about the ventricles being in diastole or systole, okay? It just sort of depends. But normally, when we talk about this, we usually use these terms just by themselves, right? Systole and diastole. When we do the cardiac cycle, that's exactly what we're doing, systole and diastole. And when we use these terms just by themselves, we're always referring to the ventricles, okay? So if I use the terms systole, diastole, you always assume I'm talking about the ventricles, right? If I'm talking about the atria, I'll say atrial systole, atrial diastole. But if I just say the term by itself, always the ventricles. Why the ventricles? Why would I care more? Why would I care more about the ventricles than I care about the atria? What? Say it. What?
I'm just saying that's what the blood goes so it can contract and push it out.
Right, the ventricles push the blood out of the heart, right? So the ventricles are doing the heavy lifting for the heart because they're pushing the blood out, right? Either via the pulmonary trunk or the aorta, right? So when we talk about function of the heart, we generally are kind of focused on the ventricles because I know it's a little biased of us to say, but they're kind of the more important chamber because they are the ones pushing the blood out of the heart. Okay. So that's usually where we focus our energy, is looking at the ventricles. Because we care about how much blood is getting pumped out of the heart. All right, so let's go ahead and take a look at this. So there's obviously multiple ways of doing this, but most books start by putting us here in the phase of the cardiac cycle, mid to late diastole, right? So again, mid to late diastole tells us that the heart is relaxed, okay? And when it's relaxed, we have blood flowing from the superior and inferior vena cava into the right atrium, okay? And that blood is flowing into the right ventricle. Similarly, on the left side of the heart, we see the blood is flowing in from the lungs via the pulmonary veins, excuse me, and again flowing into the left ventricle, right? So this is really like just filling up a glass of water, right? The blood is just pouring into the heart, and gravity's doing most of the work here, right? As well as a little bit of pressure, but when you're standing upright, gravity's doing a lot of the work. And those atrial ventricular valves, okay, those atrioventricular valves are these loose flaps of tissue. So when the blood is flowing from the atria to the ventricles, they just kind of fall open. Okay, then we just fill the ventricle and the atrium right up with blood. Okay. At the very end of diastole, okay, at the very end of diastole, we see that the atria are going to contract. Okay, so here we see that we have atrial contraction. The atrium contracts, the atrium contracts. All right, and what that does is it pushes a little bit of extra blood into the ventricles. Okay? Remember, the ventricles, like all muscle, are made up of overlapping actin and myosin fibers. And what we're doing is we're just spreading them out a little. Okay? Just like we stretch a water balloon and we put a little bit of extra water into it, we're doing that with the ventricles. Those atria contract and they push a little bit of extra blood into your ventricles. What that's going to do is it's going to allow your ventricles to contract more forcefully. Okay? We'll talk about that a little bit next week if we have a chance, but we know that when we stretch out a sarcomere a little bit, what happens is that it's able to contract more forcefully. One of the things that we do care about is the forcefulness with which the heart contracts, right? We don't want it to be too high or too low. We want it to be, as Goldilocks found, for just right. Okay? So the atria contract, they just stretch out those ventricles a little bit, and then the ventricles contract. And we enter the next phase of the cardiac cycle, which is systole. Okay? In systole, we have two primary phases. We have the isovolumetric phase, and we have the ejection phase. In the isovolumetric phase, that word isovolumetric means same volume. So here, we don't actually eject any blood from the ventricles. What we're doing is we're pushing on the ventricles and we're popping shut those atrioventricular valves, and we're getting ready to open the semilunar valves. Okay? So this is really a phase in which we just kind of prime the valves, is basically what we're doing at this point. And then, once the atrioventricular valves are shut, we push those semilunar valves open completely, and we push the blood either into the pulmonary trunk or into the aorta, and we push blood out of the heart. Okay. Once we're done with that, the heart relaxes, and we enter diastole again, and those chambers begin to refill. Okay. So what we see here is we see a similar pattern to what we've been talking about today in terms of action potentials, right? We see that the atria are the first to contract. Okay. So what happened right before this? The SA node fired, right? It sent an action potential through the atria, and the atria contracted in response to that. As that's happening, that action potential is traveling down to the AV node, which is right about here, and that action potential spreads through the ventricles, and we have the ventricles contracting next as a result of that. Okay? So we can follow that electrical signal through the heart and see that the chambers contract as we would expect them to contract. Okay with that? Okay. So our cardiac cycle is really just an alternating cycle of diastole and systole. Right? Diastole, systole, diastole, systole. Real quick, just want to point one thing out there. You'll notice that if we look at what's happening in the heart, it's happening equally in both sides of the heart. Remember, the right side of the heart is primarily responsible for sending blood to the lungs. We talked about that on Wednesday. And the left side of the heart is primarily responsible for sending blood to the body, right? The systemic circuit. Right? We have those two circuits. If we didn't have lungs that we needed to send blood to, we would only need one atrium and one ventricle. That's how fish hearts work. They only have one atrium and one ventricle because they don't have lungs, so they don't have to worry about that. But you and I are air-breathing creatures, and we need those. We need those lungs to do their job. So we have a four-chamber heart. Anyway, it's happening equally on both sides. Okay. Next week, I'm going to finish talking about the heart, and I'm going to talk about a concept called cardiac output. Okay. It's exactly what it sounds like, though. It's how much blood your heart pumps in a given period of time. Okay? This tends to range from about 5 liters of blood per minute to, if you're exercising, up to like 30 liters of blood per minute, which is a shit ton of blood, right? I mean, that's 30 of these in 1 minute if your heart is pumping. That's a lot of blood. And we'll talk more about that whole concept next week. But it's not really, confusing, I don't think. It's just how much blood does your heart pump in a minute. So let's say, for the sake of argument, that I wanted to be able to pump 5 liters of blood per minute out of my aorta to my body. Okay? If I want to maintain that cardiac output of 5 liters per minute, how much blood do I have to have going into my right atrium every minute? 5 liters. Yeah, not a trick question, right? 5 liters. If I want to maintain a cardiac if I want to pump 5 liters out, I better have 5 liters per minute coming in, right? If that drops to 4.5 liters per minute coming in, then this drops to 4.5 liters per minute going out, right? So these two sides of the heart, the pulmonary circuit and the systemic circuit, have to be like exactly matched to each other. If they aren't, then we run into a problem, right? So the same thing is happening on both sides of the heart. And we'll expand on that concept a little bit more next week, but you get it, and that's the important part, right? It's one of the nice things in this class that makes perfect sense. Okay. So, we have the diastole and systole. Okay? This is following the contraction of the heart. One of the things that we often, like the terms that we often use to describe this, is we often call these the mechanical events of the heart or the contractile events of the heart. Different people will use different words, but when we talk about something happening mechanically, it's moving, right? So this heart, this cardiac muscle. So let's go ahead and take a look at another way in which we could sort of map this. And then we'll relate it back to this. Probably the easiest way for us to sort of map this or look at this or visualize this, whatever verb you want to use, is through something called an electrocardiogram. You guys have experience with ECGs. No? Ever had one done? No? Yeah. All right. Okay, cool. All right, cool. Here you guys do an electrocardiogram. ECG. Sometimes they're called EKGs. Really the same thing. It's just that EKG uses the German spelling of cardio, which is with a K. The Germans use the cardio with a K, right? It's a terrible German accent. It sounds really bad. But nevertheless, it's the same thing. So the term EKG and ECG are really used interchangeably. Right? What is an electrocardiogram? Well, think about the word, right? You see cardio in here. That means heart. Okay? And gram, a draw a typical ECG trace on the board. We'll kind of go through what it means, and then what we'll do is we will correlate it to this. Put this diagram back up. Put an ECG trace here. I'm going to draw 2 cycles, 2 heartbeats, just so that we have something here to sort of see how it kind of repeats itself. Something along those lines. Not perfect, but it will do. I've repeated it twice. Okay. So in an ECG trace, we have a couple of really notable features. The first one that we have is right here. This one is called P wave. We have our second P wave over here. Okay. The P wave represents the depolarization of the depolarization of the atrium. So remember, in our cardiac cycle, the first thing that happens is that the atria depolarize. Right? The SA node fires, and we have the depolarization of the atrium. Okay. The next thing that we see, I guess I should draw it. Make them a little bit more equal. I didn't mean to make them so unequal. I'm not a good artist. I think I will do this now. Q, R, S, right? So the next 3 waves that we have are what we usually just call the QRS complex. But they are 3 separate waves. Okay? So again, over here we have Q, R, and S. QR and S. The QRS complex is the represents the depolarization of the ventricles. Depolarization of the ventricles, the QRS complex. Now, this makes sense when we look at it, right? Because if I compare my P wave to my QRS complex, I see that the QRS complex is way bigger, right? Way taller. Well, that makes sense because if I compare the size of my atria to the size of my ventricles, I see that there's a lot more tissue here to depolarize in my ventricles, right? So it makes sense that it would be a bigger electrical event. Okay. And then finally, last but not least, we have this guy, and these should also be equal. Don't judge me too hard. The T wave over here. Okay? Unless I'm giving my guy here some sort of cardiac value, which I'm not trying to do. We have the T wave, which represents repolarization of the ventricles. Repolarization of the ventricles, right? If it depolarizes, it's got to repolarize, right? So we have that. So this is a typical ECG trace, right? We have the P wave where the atria depolarize, QRS where the ventricles depolarize, and then we have the T wave, which is the repolarization of the ventricles. What are we missing? Repolarization of the atria, right? When would that take place? So if I've got, with my QRS, if I've got depolarization, repolarization, with my atria, I've got depolarization, like right here, right? The thing is the QRS complex is really big. That means we're talking the ventricles are big, right, compared to the atria. So what happens is that the atria are actually repolarizing right in here. We just don't see it in the ECG trace because it's kind of hidden behind the QRS complex. So we just can't see it, but it is happening at that point. Yeah. All right. So the other thing then that we can do, so notice that in these descriptions, the P wave electrical events. Okay, we're following the pathway that the electrical charge takes, or the electrical disruption takes through the heart. Okay? But you guys, as educated physiology students, know that what happens after the depolarization is what? Well, yes, but what does a muscle do when it's depolarized? Contracts, right? It shortens. So you guys know that the next thing that's going to happen after a depolarization is a contraction, right? The P wave is not representative of contraction, okay? So when I'm going to quiz or an exam, I'm going to ask the question, what does the P wave mean? Don't tell me it means the atrial contraction, because it doesn't mean that. It's depolarization. This is a graph of the electrical events of the heart. But we can map the mechanical events, the contractile events, onto this, right? So we know that when a muscle is depolarized, we know that when a muscle is depolarized, we know it's going to contract. Okay? So let's go ahead and do that. So here we are depolarizing the ventricles. It turns out that the ventricles start contracting right about here. Okay? Start contracting right about here. So this is where ventricular systole begins. Okay? And it goes until about 1/3 of the way down the ventricle. So it goes until about there. So this right here would be ventricular. And then the diastole, relaxation, would take place all the way to the peak of the next R wave. And then we would start again. Okay? So again, systole, diastole, systole, diastole, and so on and so forth. Okay? There's a whole bunch of things that we can detect from an EKG. Okay, and again, this is going to be the first order of business. When you go into the emergency room and you're complaining about heart issues, the very first thing they're going to want to do is they're going to want to see blood work, but they're also going to want to see a picture of what's happening electrically in your heart. Okay, why? Because if you have, you're skipping waves, or if you have an elevated wave, or a long time between these things, it could indicate you have a blockage somewhere because you have dead or dying tissue somewhere in your heart. You could have blood flow that has been blocked somewhere, and we have the heart not contracting or something like that. So you should see a really easy flow of electrical information from thing to thing to thing in the heart. And if that's disrupted, that can be indicative of all sorts of different problems. There's a book on EKGs and interpreting abnormal EKGs that's about that thick. And I admittedly don't know much about it. It's a little bit out of my wheelhouse. If you go into an EMT class, or if you go down the route of working on hearts, you're going to have to learn all of that. And it's really interesting stuff. I think at the end of the day, I think it's all pretty logical, but there's definitely a lot there, and there's different things that we can see happening and stuff like that. Lab 12 does give you guys a couple of abnormal ECGs to kind of take a look at and try to interpret. And after today, hopefully you'll be able to do that. But what I want to spend kind of the rest of our time on today is having us do our own ECGs on ourselves. And let's go ahead and kind of take a look and see what's happening, right? So let's do that. Let's take a minute. And we're going to need some desk space. You have everything that you need on your desk, but maybe if you have something out that you're not going to need today, put it away.